Hello everyone, my name is Ivan and I'll be explaining HIV and AIDS. So for the introduction, HIV stands for Human Immunodeficiency Virus and AIDS stands for Acquired Immune Deficiency Syndrome. In 1984, Thailand was the first Southeast Asian region country to report a case of HIV. HIV originated from chimpanzees that are infected from SIV, which stands for Simian Immunodeficiency Virus. HIV was first identified among the homosexuals in the US at 1981. Further information on HIV includes an incubation period that varies from a few months up to 10 or more years. After 2 to 4 weeks of contracting the virus, there is a large amount of virus in their blood, which makes them very contagious. So for the structure of HIV, there is a capsule-like structure called the HIV capsid that's located in the center, and the HIV RNA and the HIV enzymes are located inside the capsid. HIV RNA contains genetic material and HIV enzymes are proteins that carry out steps in the HIV life cycle. On the surface is the HIV envelope and the glycoproteins, which are embedded spikes on the envelope. There are 7 stages in the life cycle of HIV. For the first stage, which is integration, HIV releases and uses integrase in the CD4 cell to insert its viral DNA into the DNA of the CD4 cell. The second stage, replication, is when HIV uses the machinery of the CD4 cell to make long protein chains of HIV, which are the building blocks of HIV. For the third and fourth stage, which is assembly and budding, HIV proteins and RNA assemble on the surface of the CD4 cell, and budding is when immature HIV pushes itself out of the host, and protease is released to break long chain of protein to create mature virus, which is infectious. For the fifth stage, which is binding, HIV binds itself onto the receptors on the surface of the CD4 cell. And for fusion is when the HIV envelope and the CD4 cell membrane fuse together, allowing HIV to enter the CD4 cell. Reverse transcription is when in the CD4 cell, HIV releases and uses an enzyme called reverse transcriptase to convert HIV RNA into HIV DNA. HIV can be transmitted through blood, bodily fluids, except saliva, unprotected sex, and sharing needles. There are 3 phases displayed in the graph. The transmission potential is a quantitative indicator of the epidemiological efficiency of infection with a given viral load. The mean amount of predicted secondary infections over the whole chronic and asymptomatic infectious lifespan, calculated to be the result of the infection and the length of infection. Highlighted in yellow, the HIV-1 infection has a very strong rate in the first weeks of infection and a steady spike in the late stages of infection. Displayed in gray is the infectiousness measured in married individuals. The extent of the virus calculated to be the median duration from seroconversion to AIDS is displayed in green.